Good morning, students. I am Dr. Abhishek, Tutor, Department of Physiology, and I am continuing with the chapter of Central Nervous System. Today, we'll discuss about the topics which is we are continuing synaptic transmission, and in that synaptic transmission, the last topic which we are going to discuss today is neurotransmitter, about which we were discussing throughout the last few classes. So we'll start and we'll discuss about neurotransmitter today. So, starting with the neurotransmitter, we have been listening about this neurotransmitter since we have been studying synaptic transmission in the last few classes. So, today we'll see what actually these neurotransmitters are. They are the chemical substances liberated at the nerve endings that helps to transfer message in form of nerve impulse from presynaptic to postsynaptic neuron. We already know very well what are presynaptic neurons and what are postsynaptic neurons. So these neurotransmitters are the chemical substances that helps in transfer of the messages in form of nerve impulse. Now they are released from neurons supplying muscles or a gland. When they are released, they also circulate as a hormone, and they are therefore also called as neurohormones. For example, norepinephrine, ADH, that is, and gonadotropin releasing hormone, antidiuretic hormone, and gonadotropin releasing the hormone GnRH. So these are the examples of neurohormones, and they are again neurotransmitters. Now, what are the characteristics which makes a chemical substance to be a neurotransmitter? First, it must be synthesized in the neuron concerned. Second, it should be stored in the presynaptic endings. Third, it should be released at the synapse. Fourth, it should have a specific receptor on the postsynaptic membrane. And fifth, it should be disposed as soon as action is over. So these are the 5 things which makes a chemical substance a neurotransmitter. Then the diagram which is shown on the slide here is a classification of these neurotransmitters. So based upon their chemical structures, you can see these amino acids can be peptides, can be biogenic amines, and can be acetylcholine. So based upon the chemical structures, they are in amino acids. The examples are aspartic acids, glycine, glutamic acids, and GABA. That is, GABA is, the full form of GABA is gamma aminobutyric acid. Then in peptides we have vasopressin, somatostatins, and neurotensine. And in biogenic amines we have norepinephrine, dopamine, they are together called catecholamines, and serotonins. And acetylcholine alone itself another neurotransmitter, which is basically one of the most important neurotransmitters which are present in many parts of the nervous system. So it is a excitatory type. So the neurotransmitter can also be classified based on this is another way of classifying the neurotransmitter, what we have just talked. Class 1 was acetylcholine, class 2 includes biogenic amines, that is catecholamines, epinephrine, norepinephrine, dopamine, serotonin, histamine. Class 3 group includes amino acid neurotransmitters, which is GABA, gamma aminobutyric acid, glycine, glutamate, aspartate. Then class 4 is nitrous oxide and even carbon monoxide. And then neuropeptide transmitter includes neuroactive peptides, pituitary peptides. Neuroactive peptides are ADH and oxytocin. If you are studying endocrinology, you must be very well aware about these ADH and oxytocin. These are called neuroactive peptides. Then pituitary peptides, which includes CRH, and somatostatins, even gonadotropin releasing hormone. Then peptides acting on gut and brain, and then neuropeptides from other tissues. So these are another way of biochemically classifying the neurotransmitters. If you get a short course on a neurotransmitter, you have to very well describe what the neurotransmitters are, and then you have to classify and give examples of the classification as it is shown here in the slide. Then we were talking about the functional classification of neurotransmitter. This is the functional classification of neurotransmitter, the excitatory neurotransmitters and inhibitory neurotransmitter. We have talked about excitatory postsynaptic potential and inhibitory postsynaptic potentials. The excitatory had excitatory neurotransmitter, the inhibitory had inhibitory neurotransmitters, and you have to know the examples of them. That excitatory neurotransmitter includes glutamate, aspartate, adrenaline, noradrenaline, nitric oxide, acetylcholine, and inhibitory types are GABA, glycine, adrenaline, even noradrenaline. There are some which have a common, that is, they have both inhibitory as well as excitatory roles. So they are categorized under both the categories, both inhibitory as well as excitatory. As you can see here in the examples, glycine is one of them, you can see here, which has both excitatory as well as inhibitory roles. So this was all about neurotransmitter. I'll just talk in very short about the one or two neurotransmitters which are very, very important in the nervous system. One is acetylcholine, which is an excitatory type, present in postganglionic parasympathetic neuron, all autonomic preganglionic neurons. They are even present in postganglionic sympathetic neurons supplying sweat glands, blood vessels of skeletal muscles. They are present at neuromuscular junctions and in amacrine cells in retina. Now, how it is synthesized? Very simple. This choline combines with acetyl-CoA in presence of choline acetyl transferase to form acetylcholine plus CoA. Now, there are two types of each neurotransmitter has a specific receptors. We talked about in the characteristics of neurotransmitters. So, the receptors here for acetylcholine are of two types: nicotinic receptors, muscarinic receptors. Nicotinic receptors have further 5 subtypes, while muscarinic receptors have another 5 subtypes. The 5 subtypes of nicotinic receptors are alpha, beta, gamma, delta, and epsilon, while the muscarinic receptors 5 subtypes are named from M1 to M5. Atropine blocks all these 5 receptors. You can get an MCQ on this. Atropine is a drug or chemical which can block all these 5 receptors. Then another important inhibitory type, this was the excitatory type we talked about, neurotransmitter, acetylcholine. Then another major inhibitory neurotransmitter in brain and in retina, in brain is GABA, that is GABA aminobutyric acid. It has 3 types of receptors, GABA A, GABA B, and GABA C. GABA A and B are present in brain, while GABA C is present in retina. Then just one clinical aspect of this GABA receptors, that benzodiazepine, a very, very commonly used drug in general practice. This drug acts by increasing the chloride conductance via GABA A receptors and thus causing sedation, muscle relaxation. It also has an anti-convulsant effect as well as anti-anxiety effect. So benzodiazepine is one of the most very commonly prescribed drugs, not only by a psychiatrist but even by the general practitioners in cases of generalized anxiety disorders. Here, alcohol and barbiturates also acts partially by increasing chloride conductance through chloride channels. So this again, here the GABA comes in role and this brings on action. Then glycine, we have already talked about that glycine is both an inhibitory as well as excitatory. It is excitatory in some parts of the brain and it facilitates pain transmission. We'll talk in details about pain. Now, this was all about the synaptic transmission in central nervous system. We were talking since last 2 to 3 classes. Neurotransmitter comes as a short notes in here. Even you can get a long question on synaptic transmission. synaptic potential. You will get a short notes on synaptic potential, long question on synaptic transmission mechanism, and you have to draw a good diagram and explain all this. Now we come to a new chapter. Today we'll start with that chapter and continue that in the next class. We'll be starting with this new chapter. The chapter name is Sensory System. Sensory System and Physiology of Receptor. There are two parts in this chapter. First is sensory system. How is the sensory system made up of? It consists of a receptor, it consists of an afferent, and it consists of neurons in CNS. Receptors are what? That receives and transduce the stimulus into action potential in the sensory neurons. Afferent. What are afferents? They are the pathways of the sensory neurons which transmit the stimulus in form of action potential to the central nervous system. And then neurons in the CNS modulate the sensory information and the areas in the brain, and that recognizes a stimulus. This is so many things are happening in the sensory system. So sensory system is made up of these three things which coordinate together, they work together to recognize a simple stimulus which you can receive through any of your body parts. Then we define sensation. What is sensation? It is a basic recognition of a stimulus. Just a basic recognition. So this is a feeling. So it's called aesthesia. It's a feeling. Another thing, absence of sensation. What is absence of sensation? Anesthesia. And when there is abnormal sensation, the term is called paresthesia. So there is aesthesia, that is feeling. There is anesthesia, that is absence of sensation or feeling. And then there is abnormal feeling or abnormal sensation, which is called as paresthesia. Then there is sensation of movement, that is called as kinesthesia. Sensation of movement is kinesthesia. Any of these can come as an MCQ. So each of these is important. Then how we categorize. So this is what we just talked about. You can see in the slide. Basic recognition, then absence, then abnormal, and then sensation of movement. Then how we classify the type. It is special, visceral, and somatic. Special, the class is being taken by Dr. Sweta, who is talking to you about the sensory organs, and the examples you already know. They include vision, audition, and gustation, that is taste. And they have specific sensory receptors present there to receive these sensations. So they all comes under special type of sensation. Then we have visceral. Visceral means the body, the internal body organs. So stimulation of the receptors in the viscera. And where are they present? They are present in the wall of the viscera. The visceral organs which are present in skull, abdomen, thorax, pelvis, they all have receptors present in their wall which receive these sensations. And the third type of sensation is somatic, also called as somesthesia, which is present in the body surface or on the body surface. Then on bones, joints, connective tissues, includes sensation of touch, pain, temperature, vibration, and even joint movement. So they all come under somatic sensation. Then what is perception? Perception is appreciation and interpretation of sensation. What all comes under appreciation and interpretation? Recognition, comparison, discrimination, and integration of sensation. So I repeat, recognition, comparison, discrimination, and integration of the sensation all comes under appreciation and interpretation of sensation, and that is called as perception. Then what is another term is there? Sensory modulation. What is sensory modulation? Either accentuation or inhibition. Either accentuation or inhibition of sensation is called sensory modulation. This is a type of a control. Either there is an increase or there is a decrease in the sensation. So this type of control is called sensory modulation. Then we come to another very important thing, dimensions of sensation. Broadly, in different books you can see lot more dimensions of sensation with different terminologies, but broadly in writing a short notes on sensation or any of these, you can broadly classify them under modality, intensity, affect and acuity. The name itself gives you the explanation. Modality means quality or the type. For example, modality of tactile sensation is touch. Tactile sensation, modality is touch. So what's the type? So that is called modality. Second is intensity. What is intensity? It is degree of perception. We know what is perception. That is appreciation and interpretation of sensation. So degree of that perception is intensity. For example, there can be rapidly adapting receptors, there can be slowly adapting, and there can be non-adapting also. For a rapidly adapting, like spontaneous decrease in the intensity quickly when the stimulus is applied. For a little longer duration, then that is called as rapidly adapting. When there is decrease in the intensity if the stimulus is applied for a little longer duration, then there is a decrease in the intensity, and that type of receptor will have, that will be a rapidly adapting sensation. For example, touch. Then there can be sensation, there is the decrease in the intensity is slow, so that is called as slowly adapting. And there can be a sensation that does not change its intensity, and it is said to be non-adapting. Example to that is pain. Then we come to the third dimension of sensation, and that is called as affect. What do you mean by affect? The emotional component of sensation is called affect. It can either be positive, negative or neutral. Positive when it evokes a pleasant emotional sensation, then it is positive affect. Negative when it evokes an unpleasant emotional sensation or response. Example, pain. So that is a negative affect. And neutral when it does not have emotional component. Affect means the emotional component of sensation. So when positive, there is a pleasant response. When there is negative, there is an unpleasant emotional response. And neutral, there is no emotional component attached to it. Then the fourth dimension of sensation is acuity. Acuity, you have also heard this term when you were studying eye. Acuity is precision of stimulus localization. How precisely you can localize a stimulus, that is called as acuity. And how it varies, or it varies in its precision. There are three components governing that. First, the concentration of receptors, that is the innervation density. Second, receptive field size of the area. What is the size of that area which is getting that stimulus? And third is the size of the cortical area representing that somatic body part. We'll talk or we'll see about this thing when we read or we learn more chapters in CNS, and we'll see how there is a cortical area representation in the brain of the various somatic body parts, how these areas are represented in the brain. So that acuity depends even on that. So there can be constant, so it varies, the acuity or the precision varies with concentration of the receptors, receptive field size of the area, and the size of the cortical area representing that somatic body parts. So that was all about the first chapter which we have started today after finishing the neurotransmitter topic, that is sensory system and physiology of receptors. In the next class, we'll talk about receptors. What are receptors? What are receptors? How we define receptors? How we classify receptors? What are the important receptors in our body? And the properties of receptors, which we'll be talking in the next class. So that's all for the class today, students. Thank you so much.